Hey there gang, and welcome to our video covering DNA replication. We are still in section 9.3 here in your biology textbook, and we're still looking at deoxyribonucleic acid. So far we've talked about the structure and composition of deoxyribonucleic acid. We've talked about the nucleotide and how it is the monomer and DNA is the polymer. And now we're going to move on and we're going to talk about how DNA makes more of itself. We're going to talk DNA replication, which you see right here on the screen right now. What do you do when you replicate something? Well, you make more of it. Okay? So what we're looking at today is the making of more DNA. Major concept for this is you want to be able to outline and describe and be able to recognize the steps of the DNA replication process and also be able to demonstrate your knowledge about the enzymes that are used in each one of those steps. Okay, so those are our goals for today here. All right. So a couple things here quickly about DNA replication. It is replicated during what we call the S phase of the cell cycle, or the synthesis phase. We'll study that here in about a lesson or two. All right, but you would have already seen it had you read chapter 6. It's the S phase, synthesis. Synthesis meaning making of, or making of something. So during the cell cycle, DNA is replicated during the S phase. Because the nitrogen bases are complementary with each other, because we have that complementary relationship, we saw that back a lesson ago, exact copies of DNA can be easily made. All right, and we'll see that here in a second. So, courtesy of the complementary relationships between those nitrogen bases, it's kind of easy to replicate DNA. Now, the actual process here, the very first step we refer to as unwinding and unzipping. You have to imagine again that DNA double helix is a twisted staircase or a twisted ladder. So in order to make more of it, we need to untwist it or unwind it and then unzip it. All right, unzipping meaning break it apart in the center, break those hydrogen bonds. The actual enzyme that's at work here is known as DNA helicase. And that heli, H-E-L-I, is really important here because remember that DNA is in a double helix. Okay? So enzymes usually end with ACE or ICE or something along those lines. So DNA helicase, and enzymes usually break down or build something up. So we're looking at something that's going to break down. Okay? So what breaks down, what unwinds and unzips the DNA double helix? DNA helicase. Okay? It actually breaks those hydrogen bonds between the two nucleotide strands. Now, those molecules, those nitrogen bases are going to want to go back to one another and attach. They're going to want to recreate hydrogen bonds. Well, we've got some specialized proteins that lodge themselves into the area between these separated nucleotide strands. Okay? The location at which those proteins lodge themselves is known as the replication forks. Okay, so step one, in review, we are unwinding and unzipping the DNA double helix. The enzyme being used is DNA helicase. Okay, how do you unzip it? The DNA helicase molecule, that enzyme latches on to the DNA double helix and it breaks the hydrogen bonds between the nitrogen bases. On to step 2 and 3 here. Get my little screen blocker up. Step 2, we call this base pairing. It's the addition of nucleotides. Okay, so we're going to be adding many nucleotides, a whole lot of nucleotides, and we have a root word for the word many. You've seen it in math before when you talk about a shape that has many sides. You refer to it as a polygon. Okay, again, we've seen poly. If you've got a large molecule that's composed of many subunits that are identical to one another, it's considered a polymer. It's many of those small subunits. So, what enzyme is going to be adding many nucleotides to the DNA double helix? DNA polymerase. Okay, if you want to call it polymerase, you may, but we correctly pronunciate it DNA polymerase. It latches on to the two separated sides of the ladder, the two separated nucleotide strands, and begins to add complementary nucleotides. Okay? So the RNA polymerase is going to latch onto that strand, read the strand, and add new nucleotides at the same time. Two new strands of DNA are formed during step 2. Step 3 is pretty simple. It's a stop sign, okay? Replication is signaled to stop. The polymerases detach, and the two new DNA molecules exist and are identical to each other, because remember that complementary relationship. Each new molecule has an old strand and a new strand. We refer to DNA replication as a semi-conservative process, okay? Because again, we're going from one DNA molecule to two DNA molecules. They're not completely brand new. Only one strand of each new DNA molecule is actually brand spanking new. One strand from each DNA molecule is old. They each came from the original strand that has been replicated. So semi-conservative. We're not conserving the entire thing. All right? It's not just being, what's what I'm looking for here? Completely redone. Okay? We're creating two new things that each have a little bit of the old one in them. So semi-conservative replication. All right? One other term I didn't use in here is template strand. Okay? So when we break that old DNA molecule in half, each one of those old nucleotide strands, we refer to them as template strands. Your book talks about that a lot. I didn't mention it yet here in the video, but we will be using it. Now, I'm going to go ahead and stop the video here. Hopefully you feel a little bit informed on the DNA replication process. Hopefully you can give a general description for these steps, and you also know on the back of your hand here what the enzymes are that are used in each one of those steps, one of those stages, one of those processes, okay? We're going to be doing some DNA replication in class. Remember, you build a DNA molecule one nucleotide at a time, one strand at a time, and I'll see you later. Bye.